balanced relationship for haplotypes. So this measure has been used in a recent context to understand long-term population estimates for Antarctic minke whales. So I can give an illustrative example here. Kristen Ruge headed this study, which was published a couple of years ago, and what they were trying to understand was what is the long-term population abundance for Antarctic minke whales. They were trying to address this question in response to a recent arguments about minke whales having increased in abundance in the 21st century as a consequence of baleen whales being killed in the southern hemisphere in the 20th century. So the idea was that when the baleen whales were killed, then all the krill that they would have eaten was available to the minke whales, and the minke whales increased in abundance. So knowing that this hypothesis suggests an increase in recent abundance of minke whales, this team tried to estimate how the abundance was prior to whaling using the theta estimate. So you need to calculate your nucleotide diversity, and they did this from using 11 intronic genes, and they used genetic substitution rate calculated from baleen whales and a generation time estimate based on whaling data to give them a large estimate of about 600 to 700,000 individuals in the Antarctic prior to whaling. You can see that the confidence boundaries are very wide on this, between 300 to 1 million, 300,000 to 1 million, because of they accounted for uncertainty in all of these parameters. But they were able to conclude from this that the current abundance of minkies is the same, if not slightly less, than it was in the past, which contradicts the theory that they may have been increasing recently as a consequence of krill release. Okay, so this slide again says basically that population changes occur through time. We can understand population changes through time using present-day molecular diversity. So using the similar reconstruction of lineages based on current lineages, genetic lineages in the population, when the genetic data is extremely variable, we can start to reconstruct how diversity has changed through the past back to the last lineage coalescence of that species. So here, the diagram here shows how we plot the southern hemisphere humpback diversity through time, going back, looking at theta diversity again, going back before present. And this analysis was done by segregating southern hemisphere diversity from the northern hemisphere diversity in humpbacks, and it's based on about 2,800 control region mitochondrial DNA sequences. And what this analysis shows is that there seems to have been some expansion in humpback diversity since the last glacial maximum, according to the arrow around about 12,000 years before present here. It's also worth noting that this blip reflects the convergence of deep lineages within the humpback species, right at the very base of the humpback phylogeny. So a point I'd make about these interesting deeper population reconstructions is that they don't go up to the present day, because the evolutionary rate calculated from mitochondrial DNA for whales is very slow. So in fact, the last point of information here in this graph of diversity through time is around 500 years ago, which is prior to the advent of humpback whaling. So we, at this point, are standing at the edge of a lot of new next generation sequencing, which is going to allow for these kinds of reconstructions using things like single nucleotide polymorphism data, will allow us to bring some of this skyline information towards into the present. But at present, it tends to reflect longer period abundance changes through time. As Phil and Yulia discussed earlier on, there were massive 20th century kills of Southern Ocean baleen whales. These reconstructions reflect a few different data sources, but the take-home message was that roughly 2 million were killed in the, well, about 1.5 million were killed in this listing just in the Southern Ocean for Southern Ocean associated whales, which is an enormous number that needs to be incorporated into population dynamic reconstructions. What's less often discussed is that the 19th century killing was also very substantial. For right whales, that was where the majority of the hits came, but humpbacks also received quite substantial catches in the 19th century. This just reflects different types of whaling that were carried out and when they first started. So this is, again, this is just the 19th century whale kills. So we know that there's been a long history of whaling for some species. And genetics interacts with population assessments in a useful way now, but it's only really just begun. So population assessments of baleen whales, as Nick discussed right at the very beginning of today, was carried out at the IWC in order to sort of carry out comprehensive assessments and understand population recovery. Genetics aids population assessments by providing a tool for individual identification through microsatellite genotypes, and it also allows us to understand habitat usage in a sex-specific way. We can understand how populations mix on shared feeding grounds when, for example, you have two independent breeding grounds and then those animals from those breeding grounds are all found on a common feeding ground. We can use population assignment identifications to understand population mixing. And something I'm going to discuss a bit more here is how we can relate haplotype diversity to minimum bottleneck size during recent whaling exploitation. Now, haplotypes are genetic lineages, mitochondrially inherited genetic lineages. So mitochondria is inherited through the mother, and so every unique mitochondrial lineage represents at least one mother in the population. Population dynamics models are used to understand how whale abundance has changed in response to whaling. This shows a simple model which was done to understand the population history of southern right whales in 2001, and the trends basically are from 1770 to the present day, and the population dynamic model needs to take into account the current abundance and trends in abundance and the catch history. The population model tries to find the initial abundance value here that most closely fits estimated current trends in abundance. This has been a standard model which has been used for a number of years at the IWC, and most recently the models are being used to try and to include minimum bottleneck abundance as a constraint, and also to account for population substructure, where you have populations that exchange migrants during the course of exploitation and prior to exploitation. These models are quite simple, and they also assume a static carrying capacity through time. So the minimum bottleneck abundance is quite a crucial thing, because as you can see from this slide, the minimum number of whales estimated for the whole southern right whale population in the entire southern hemisphere was 60 females, according to a standard IWC model in 2001. This is based on a growth rate of about 7.5%. That's extremely low. We took a piece of information which you can obtain from the current populations, which is the number of haplotypes. So if you sample a contemporary population, if you take a genetic sample of a contemporary population, then the number of haplotypes in that contemporary population gives you the minimum number of mothers that must have passed through the bottleneck, because haplotypes are maternally inherited. So this just shows time, and it shows number of animals through time. If you think that that's the minimum number of mothers that must have passed through the bottleneck, then there's going to be more animals aside from the mothers. There'll be the fathers, and there'll also be immature whales that come through the bottleneck that carry the same mitochondrial DNA. So as an absolute minimum constraint on these population models, we have started to impose a lower boundary on bottleneck size in the population dynamic models based on 4 times the number of haplotypes in a contemporary sample. The bound is a very conservative one because it doesn't take into account the fact that multiple families carrying the same haplotype lineage would have gone through the bottleneck. So in order to start thinking about how to estimate bottleneck size, we proposed a set of measures that could take you from a current genetic sample, and this is a genetic sample showing some different haplotypes in the population, and then extrapolating that to starting to get a bit of an estimate of what the minimum abundance might have been. So if you take the genetic sample, and then you're going to have a variety of frequencies of haplotypes, and you can fit this to a discovery curve of haplotypes discovered through over different numbers of sample sizes. By doing this, you can extrapolate to a population sample of haplotypes from the original sample that you have. If you take that, then you consider that some new haplotypes might be described by longer DNA sequence lengths, then sometimes it's possible to increase the population sample based on sequencing longer lengths of mitochondrial DNA also. An additional consideration is how the haplotype frequency distribution is at the bottleneck. It's not likely that you're going to get a ratio of 1 to 1 to 1. Every individual has a unique haplotype because haplotypes are distributed. There tends to be a trend between very common and then quite a few rare haplotypes in the population. So we needed to try and account for the fact that there would be a frequency distribution at the bottleneck. And one way you can do this is to estimate what the past haplotype frequency distribution may have been using the Theta measure of genetic diversity, which I discussed in the previous slide. And so picking samples from different from a past population haplotype frequency distribution, you can find the number of samples that's needed to give you your current population haplotype sample, and therefore come to an estimate of what the abundance would be given that haplotype frequency distribution. A more problematic thing to consider is how haplotypes are lost since the bottleneck, because it's likely that animals that went through the bottleneck died and their lineages died with them. But it's actually a rather difficult thing to model. But all of these aspects basically speak. They speak to individual-based models better than to this traditional population dynamic model, which was originally used to reconstruct population histories. We also need to consider that males and immature animals are a significant component of the population that passes through the bottleneck, and information on that can be got from life history analysis of the animals in the present day and looking at whaling data from the past. We have used these metrics, which in a very conservative framework up to this point, to try and understand, if you look at the most conservative values of N-min, when you take the absolute minimum that might have the minimum haplotype numbers and try and be very conservative about your upward correction, then we find that the minimum bottleneck size it still tends to fall above the estimated abundances for southern right whales here. Now this is the southern right whale trajectory I showed you before, and this inset shows abundances between 1870 and 1970, and this green line shows the base case scenario for population abundance, and the N-min value calculated using the corrections on the previous slide is way above the estimated minimum bottleneck size, showing that N-min effectively is higher than the population dynamic model would suggest. More recently, slightly different methods were used with the same general intent to estimate what the N-min number would be, again very conservatively for blue whales, and it fell very close to the estimated minimum abundance based on population models calculated by Trevor Branch. As Scott showed in one of his slides earlier, Angie Srember's work has updated the number of haplotypes that are now known for blue whales. So these show the number of haplotypes from each current population and then what the N-min calculation was. We now know that the number of haplotypes in the blue whale population is at least 52 based on calculations, so the N-min value is likely to get much larger. This is really a work in progress in terms of trying to understand how current genetic samples can enable us to estimate bottleneck sizes. We're getting closer, but we need to use new methods because these population dynamic models at the moment are fairly inflexible to imposing lower boundaries on them. So the approximate Bayesian computation methods provide one avenue by which we might be able to start incorporating genetic data more directly into the population models. Thank you. In the future, we know that carrying capacity in the Southern Ocean is likely to be changing. Krill abundance seems to have declined in the last 50 to 100 years. We also know that the whaling, whilst it's been very substantial in the 20th century, may have had longer term impacts, and that's not always considered in some of these models, such as the humpback models, IWC models, which just show 20th century whaling. We have a sense that things are definitely changing and that we need to understand how population dynamic models are going to, how population dynamic models will be better fitted to changing carrying capacity in the Southern Ocean. So genetics is going to be integral to that. It allows us to, first we've understood the baselines for a number of the large baleen whales, but we're going to be using these genomic markers to detect changes in population abundance and connectivity over time through mark recapture and mixed stock analysis. It will also be informing minimum abundance, and obviously as well as allowing us to understand a lot more about population change at a demographic level. We can use all the samples that we collect to understand feeding locations and cetacean health and pathogen loads in concert. Thank you.